I am Dr. Richa Gupta. I am your faculty for physiology in Frontal Lobe. And in Frontal Lobe, we have various aspects to prepare you for exams. We have previous year question based lectures where we will not only discuss the previous year questions, but we will also target high yield questions from that topic also, so that any question which comes from that topic you are able to solve. Then there are FMGE level test series, so as to prepare you for exam stress. So there is mock exam consisting of exactly the same conditions as that in exam, so that if you are prepared for the subject, another aspect is your mental stress level, so that also you are trained with. Then we have doubt and strategy sessions where you will have interactive sessions with faculty, and you can have direct support from faculty. And then also there is a last minute revision booklet, so as to prepare you for that charged revision during the last 7 days of exam. So with this, let us start with a revision of respiratory physiology MCQs. So first is, what is the primary function of surfactant in the lungs? So what is surfactant basically? Surfactant is a substance which is secreted from type 2 pneumocytes in the lungs. Okay, so if this is the alveoli is lined by two types of cells, type 1 and type 2, and type 2 pneumocytes, which are cuboidal in nature, cuboidal in structure, they secrete this surfactant. And actually wherever there is air and there is lining of water, all the alveoli there is lining of water, so at this air and water interface there is pulling, always there is pulling, there is a force which pulls. So this force is known as surface tension. Okay, so this force will tend to pull the alveoli together, and this will cause the collapse of the alveoli. Now this is prevented by what is known as surfactant, which is secreted by type 2 cuboidal cells. So what is the function of surfactant? It decreases the surface tension, and hence it decreases the alveolar collapse. So basically, by preventing the decrease in the alveolar collapse, it will help in the expansion of the alveoli. It is promoting the expansion of alveoli. Okay, and what is that? That is known as compliance. Okay, compliance. So basically, surfactant is increasing the compliance. It is increasing the stretchability of the alveoli, and with increase in stretchability, if it is supporting that, there will be decrease in work of breathing. Okay, so all these options can be part of your MCQ, where they can ask it decreases alveolar collapse, it increases compliance, it decreases the work of breathing. Any of this can be asked. Now because it is decreasing the collapse, it also prevents pulmonary edema. Very important. Okay, because what happens that with this collapse thing, when it is happened, here blood vessel is there, blood vessel. Okay, so there will be movement of the water as well because of this pull force. So since it is preventing this collapse, this pull force is decreased, and hence pulmonary edema is also prevented. Okay, so let us see this question. What does it say? So what is the primary function? Primary function is to decrease surface tension or reduce surface tension and prevent alveolar collapse. Right? Then next question. It is about the oxyhemoglobin dissociation curve. So what does it say? Oxyhemoglobin dissociation curve. Well, oxyhemoglobin dissociation curve, it is the relationship between the partial pressure of oxygen with the percentage hemoglobin saturation. Percentage hemoglobin saturation, and it is basically an S-shaped curve, or also known as sigmoid curve. Right? So as the partial pressure of oxygen increases, percentage hemoglobin saturation increases, but it is not linear. Okay, it is sigmoid shaped, and at the partial pressure of oxygen, that is approximately 27 millimeter mercury, where the hemoglobin is 50% saturated. If it is 50% saturated, that partial pressure of oxygen is known as P50. P50 is the partial pressure of oxygen at which hemoglobin is 50% saturated. Right? Now, there are certain things which you should know here. This graph represents the affinity of the hemoglobin with the oxygen. Sometimes this graph may shift to right side, and sometimes this graph may shift to left side. When the graph is shifted to right side, this is basically decrease in the hemoglobin affinity to oxygen. That means less oxygen is going to bind to hemoglobin at the same partial pressure of oxygen. So we see at 27 millimeter mercury, here we saw that 50% of the hemoglobin is saturated. But here what we see that it is saturated less. That means binding of the oxygen to hemoglobin is less, or in other terms we can say that release of oxygen is more. Release of oxygen is more, right? So that is right shift. Now what are the conditions which can cause right shift? It is increase in carbon dioxide, increase in hydrogen ions, or both of these are acidic. So basically it is causing decrease in pH. Decrease in pH will also cause a right shift. Increase in temperature and increase in 2,3 BPG. Okay, so these will cause the right shift of the oxyhemoglobin dissociation curve. What will cause the left shift of the curve? Left shift of the curve. Left shift is basically increase in the hemoglobin affinity to oxygen. So with increased affinity means there will be decrease in the release of oxygen from the hemoglobin, more binding of the oxygen to the hemoglobin. And when will this left shift occur? It will occur in the opposite cases, that is when there is decrease in carbon dioxide, decrease in hydrogen ions, or this is what? This is decrease in acid, that means increase in the pH. Okay, so alkalosis will cause a shift in the dissociation curve to the left, or there is decrease in temperature, or there is decrease in 2,3 BPG. Okay, so all of this is the questions which can come of oxyhemoglobin dissociation curve. Now P50, what we have seen, P50, that the partial pressure of oxygen at which hemoglobin is 50% saturated. Now this 50% you see here, for the right shift it has gone here. Just extend the line. That means the partial pressure of oxygen has increased. So P50 increases with right shift of the curve, and P50 decreases with left shift of the curve. So P50 increase is a sign of decreased affinity of hemoglobin to oxygen. Okay, let us see our question. In the context of oxyhemoglobin dissociation curve, which of the following causes a shift to the right? Okay, now you can easily tell. Well, it is increase in 2,3 DPG, that we have told 2,3 BPG. It is same as 2,3 DPG. Okay, so here increase in PCO2 will cause shift to right. Increased pH, that is alkalosis, it will cause shift to the left. Decreased temperature, again shift to the left. Decreased PCO2, again shift to the left. And it is a very important concept, very commonly repeated as well in various forms. Okay, so that was about the transport of oxygen. Now moving on to questions on transport of carbon dioxide. So here is the question that which of the following describes a chloride ion exchange in red blood cells where bicarbonate ions are exchanged for chloride ions? So say this is the blood vessel, right, and this is the red blood cell, magnified version we are drawing within the blood. Now carbon dioxide, which it enters from the cells, it will enter into the blood, and then it is going to enter into the RBC. So this is RBC, it enters into the RBC, and in the RBC it combines with water in the presence of an enzyme carbonic anhydrase. So carbonic anhydrase, very important enzyme present in RBCs. Now once it combines, it forms H2CO3, and this H2CO3 dissociates into H plus ions and bicarbonate ions, right? Now this bicarbonate ion enters into the blood in exchange of chloride ions. So chloride enters into the RBC, bicarbonate moves out, and this is happening by a transporter chloride bicarbonate exchanger or bicarbonate chloride exchanger. Okay, so this concept where bicarbonate is moving out and chloride is moving in, this is known as chloride shift. Chloride shift. Okay, and that's what they are asking, that this chloride exchange in red blood cells where bicarbonate ions are exchanged, this is known as what? This is known as chloride shift. Another name for this chloride shift is Hamburger effect. Okay, so this is simple question, direct question. Chloride shift, also known as Hamburger effect. Certain other concepts we'll just touch upon here. That one thing is that we have